Hi, we are going to talk today about protein folding, but before we get to that, let's just do a quick review versus what we've been talking about. You know that we've been talking about the DNA in the nucleus and how it undergoes transcription to produce messenger RNA, which is involved then in translation here. You'll remember we just did that the last lesson. The mRNA contains triplet nucleotides, and the nucleotides are grouped in threes at a time to give a code, which we call a codon. That codon signals for a specific amino acid, and that whole process is facilitated by these tRNA who have anticodons, and they are basically the code goes from the mRNA to the tRNA, and that tRNA adds an amino acid to the growing polypeptide chain, and we have a long amino acid chain. And if you remember from yesterday, where do we go from here? How do we get from this string of amino acids into a protein? So here's just that representation. Here's a long string of amino acids all hooked together, bonded together, and we get from there to this final functioning protein. How do we do that? What happens in between here and here to get our final functioning protein? So this happens in a process called protein folding. That's what we call it. We start out with our primary structure, the amino acid chain. And here's just a different way to represent it. It's the amino acids strung together in a very specific order, specific to the particular protein that is being created. And this chain folds into a secondary structure right here, or right here. That structure, the secondary structure, will be either pleated sheets or an alpha helix, a combination of both, several of both. And the tertiary structure is the next structure, the third one. Those either sheets or coils or both of them basically fold into each other to make this kind of a tertiary structure, some sort of a glob. That tertiary structure will then, the amino acids in it will interact to give us our very final product, the quaternary structure, and it is the final functioning protein. One thing to note, this happens incredibly fast. If you might think of the translation process takes around maybe a minute, well, protein folding happens in far less than a second. As a matter of fact, this is measured in nanoseconds. 100 nanoseconds right here is like saying 0.000001 second. So the entire process happens just in the blink of an eye. So I posted a few videos. They're not actual protein folding that are happening, but there's simulations of protein folding slowed way down so you can see. And I'd really encourage you to watch those. They're only a few minutes each, and they give you a real appreciation visually for what goes on, because it's such a complex process, and there are so many pieces to these proteins, and they have to be just exact in order to function properly. So it's really an amazing process. I'd encourage you to look at that. So why do proteins need to fold? It's critical for proteins to fold in order for them to be the very specific shape that they need to be to carry out their work and their function. Here's an example of an enzyme called sucrase, and it is responsible for helping sucrose break down into fructose and glucose. And what I would like for you to note is how sucrose basically fits like a lock-in key into this enzyme. If this protein, this enzyme, were not folded into its very specific shape, it would not be able to facilitate this function of breaking that sucrose down. So each protein folds into a very specific shape to allow its very specific job to be carried out. So you might wonder why we make such a big deal about proteins. It's just another molecule. Well, proteins actually are responsible for the vast majority of all of the processes that happen in cells. And life depends a huge amount on proteins. So it's really critical that they are functioning the way they need to be functioning. I gave a few pictures, representations, they're not actual pictures, of some different proteins that you're probably familiar with: hemoglobin, insulin, human growth hormone. Those are some proteins that are very important for cell function, and these are their quaternary structures. They've been folded and they're fully functioning. I also put some words up here, just different functions of proteins. They're responsible for movement, regulation, storage, hormones, antibodies, enzymes are proteins. We rely on those for structure, transport. Life would just not be able to exist without proteins. Here are some ideas for advanced proficiency. If you'd like to continue researching this, you could look into a specific protein and how its structure specifically impacts its function. You could research mutations in, pick a protein, your favorite protein, and look at mutations or consequences of those. You could find out about how a specific protein functions on an atomic level. You could look at the chemical bonds that are involved in some of the processes that go on. You could create your own graphic representation of a specific protein, and I'm not going to give you ideas. I'll let you come up with those. I'd love to see some really creative ways of graphically representing some different proteins, or explain why you chose to represent it as you did. Any of these would be great ideas, or come up with another one. So we'll see you in class. Thanks.